Today we're going to talk a little about the genetic diversity of immunoglobulins and the major histocompatibility complex, specifically looking at the major differences between specific versus nonspecific antigen receptors. First, we will look at antigen-specific receptors, with our focus being on immunoglobulins and how their synthesis creates a great amount of diversity. The entire process of light and heavy chain synthesis contributes greatly to the amount of diversity in immunoglobulins. As depicted in the picture shown here, where we can see the heavy chain, which is on human chromosome 14, and the light chains, both kappa and lambda, found on chromosome 2. Each light and heavy chain contains many specific chain regions that undergo a lot of different recombination processes. These recombination events lead to changes in amino acids, which ultimately dictate expression. Also, V or variable genes expressed in the germline contribute to the diversity of immunoglobulins. These genes form the baseline of immunoglobulin and set minimum numbers of immunoglobulins that can be produced. VJ and VDJ combinatorial association also plays a role in the great amount of immunoglobulin diversity. Any variable gene can associate with a joining gene to form a variable region of a light chain, and any variable joining or diverse gene can also join with one another on the heavy chains. Random assortment of both the heavy and light chains also plays a role in genetic diversity of immunoglobulins. Any heavy chain produced can associate with light chains that is produced, leading to a wide variety of combinations. Junctional diversity also plays a role in genetic diversity. The phenomenon occurs when VDJ recombination on the heavy chains is imprecise and amino acids at the hypervariable region change that lead to changes in the expression of the antigen binding site. Somatic hypermutation also plays a large role in immunoglobulin diversity. This phenomenon occurs when a random point mutation in the recombined VDJ unit of an antibody's heavy or light chain variable regions gene, changing the production of amino acids, thus changing expression. This occurs 10,000 times more often than general point mutations in other areas of the chromosome. Affinity maturation also plays a role in diversity. B cells respond to antigen in the germinal center of the lymphoid tissues, which leads to somatic hypermutation, which in turn causes an increased binding affinity by antigen in antibody. Somatic gene conversion also plays a role in diversity. It is still not well studied, but it appears that in certain animal species a sort of non-reciprocal exchange of sequences is occurring. Finally, AID, or activation-induced cytidine deaminase, plays a role in class switch recombination that ultimately leads to part of DNA being removed by enzymes affecting mostly Ig variable regions, or immunoglobulin variable regions. Ultimately, there are many different DNA level factors that lead to genetic diversity of immunoglobulins and TCRs alike, and are only able to produce one type of receptor per cell. Next, we will look at what causes genetic diversity in the major histocompatibility complex and the human leukocyte antigen, which is just the same thing as the MHC, but is the human version of that. The MHC is composed of 3 different major molecule classes. The great amount of diversity here is occurring at the gene level rather than the DNA level, like we just spoke about with immunoglobulins. The MHC has a great amount of polygenicity, meaning that class 1 and class 2 molecules are coded by the multiple independent genes. Class 1 and class 2 molecules have 6 different genes that each encode for those molecules. Polymorphism is yet another gene-level force contributing to the great amount of genetic diversity, meaning multiple forms of the MHC gene exist in the population and different alleles are present in different individuals. No 2 random individuals have the same haplotype. These haplotypes, or sets of MHC class 1 and class 2 genes on an individual chromosome, are passed as a unit to each individual, meaning that each individual gets 2 genes from mom and 2 genes from dad. These class 1 and class 2 genes are codominantly expressed, meaning that the genes are transcribed from both the maternal and paternal chromosomes. These genes are also coordinately regulated, meaning that they can be expressed on a single cell at the same time. As we can see, this varies greatly from antigen-specific receptors in that the diversity is occurring at the gene level and multiple receptors can be expressed. And here you can see the polygenicity being displayed here in this map, and two different haplotypes being shown here. This concludes my presentation of genetic diversity in immunoglobulins in the major histocompatibility complex.